So good morning to everyone. This is Thursday morning, 10 a.m. Central Time in Houston. It means Enjoy Science time. Thank you all for being here. So Enjoy Science is for you, the large family of MD Anderson from all over the world, in which wonderful scientists and clinician scientists from the institution are presenting the research. I want to thank very much to you for being here. Now it's start of a new year. Happy 2022 to all of you, and hoping in all the best for everything what you are doing, and thank you for being with us. So it's a great pleasure today to start again the Enjoy Science with a wonderful and very interesting talk by Dr. Weni Wang. And I want to thank her for being here with us. And I want to thank also to the co-host Alessandra Ferraioli for being here with us and taking good care of this session. And of course to Emily Lang, who is taking over of organizing it and helping us in all the steps, and to Liz Kent for what she has done for quite 2 years for Enjoy Science. Why I'm happy to have today Weni as a speaker, it's very simple, because she is a very bright young scientist. It's exactly the type of scientist who is doing science, opening the door for completely new concepts. He has, of course, a wonderful training. She is coming from a famous Chinese university, Fudan University. Then she has done the master in Columbia, the PhD in Johns Hopkins, postdoctoral fellow in Stanford, and faculty in MD Anderson. Sincerely, I don't know what other better places to train as a bioinformatician and scientist doing research for cancer patients can be. And in fact, her publications are all in wonderful journals, are not only about deep sequencing and large data, but are focusing on important aspects of cancer, including, for example, hereditary cancers like Li-Fraumeni, which certainly are the type of cancers who can get new paradigms in cancer. As you can see from the screen, her talk from today, it's very interesting. It's speaking about something completely new, and I prize Weni for her creativity and imagination. And with this, Weni, thank you very much for being with us. Please run the show. Thank you again.
Wow. Thank you, George, for the wonderful introduction. Yeah, I'd like to thank George and Alexandra for inviting me here to give this talk at this wonderful webinar series and tell everybody about the fun science that my lab has been doing over the past few years. So without preaching to the choir with this audience, I'll just start by saying that cancer transcriptomes and genomes can vary greatly within and across tumors, and that's what we're studying. And for example, so I'm collaborating, one of my collaborators, Dr. Anna Aparicio at MD Anderson, is working with me to study the androgen-indifferent prostate cancer. So here on the left are images of 2 tumors that are collected from the same patient with 1 year apart. So the first tumor showed androgen receptor negative, but after a year, it turned to an AR positive. And both tumors maintained the same TP53 mutation status, suggesting a common cancer cell of origin. So this type of transition or change in the cancer cell transcriptome is a characteristic of plasticity. And the type of the cause of the transition can be due to the genetic alterations or treatment, or simply a stochastic transition between the states for a group of cells with the same genetic background. So as an example of the androgen-indifferent prostate cancers, understanding the trajectory of these changes to be able to enforce or identify the cells located in a favorable state that responds to treatment seems to be a plausible path to curing cancer. So the definition of plasticity is, when you talk to 1,000 researchers, you probably get 1,000 definitions. But in the literature, traditionally, it is closely linked to the cancer cell stemness or cancer stem cells. But more recently, so that concept has been going on for more than 4 decades. But over the past 10 years, the single-cell sequencing technology and its application to profiling tumor samples has introduced a more conceptual understanding of plasticity and broadened its interpretation as simply changes in genomes or transcriptomes of the same group of cells. So our central hypothesis of this talk is that a fuzzy biological concept that is improving and changing based on the technology that is being used may also be improved by mathematical definitions if we consider mathematics, as a computational biologist, as a major technology tool. So back to the example of androgen-indifferent prostate cancer. So of course, Anna and I, the first thing is we perform single-cell RNA sequencing and DNA sequencing profiling of these individual tumors to look to delineate plasticity. But at the same time, what Anna really wants is that when she runs trials, for example, this famous Dynamo trial for prostate cancer, where prostate cancer patients are divided into groups based on their initial response to ADT, is to really devise an even better trial or treatment plan for patients based on their level of plasticity or tumor heterogeneity. So how do we bring then the single cell studies or understanding of heterogeneity to the patient level? So here I would like to introduce an analogy of an iceberg. So this we can consider the single cell profiling as studying the bottom of the iceberg where we get the full view. But it is costly and so far the resources are limited to evaluating a few bottom of the iceberg at a time. Whereas bulk sequencing can be considered as scanning the tip of the iceberg, and we have much better ways to scan through all of the tip of the iceberg in the entire ocean. And that will also allow us to associate directly with other clinical data, such as 10-year or 20 years of follow-up, which is not currently feasible for single-cell sequencing profiling of the patients because the single cell is only introduced in the past 10 years. So our second hypothesis is then to scanning through the tips of the iceberg or studying the bulk sequencing data across the ocean is informative and will complement investigating just a few icebergs in full length, such as using single-cell sequencing. So why is the quantification of the bulk tissues the bottleneck? So that goes back to tumor heterogeneity. So here are two zoom-in views of one H&E slide of a resected tumor sample used for pathologist report. So on the top view, we can see that these large cells, ugly looking, and then some having a lot of open spaces suggestive of open chromatin state, are the tumor cells. And these better looking elongated cells are the fibroblasts, and then the much smaller cells with usually darker staining and solid staining are the immune lymphocytes. We can see the distribution of these different cell types are quite different between the two views, which are from the same slide, but at different spatial location. At the bottom view, the cluster of lymphocytes seemingly attacking the tumor cells, and with the tumor cells in the neighborhood presenting less open chromatin space as compared to the tumor cells in the top view. So in conclusion, the sequencing data generated from bulk tumor samples are measuring a mixture of signals from different cell types and cell states. And the compositions of these distinct cell types and states vary largely across different samples from the same or different patient. All right, so our main goal here is to study plasticity in the broadest definition, which are changes in genomes and transcriptomes of the same group of cells using deconvolution models. and to help define features of plasticity in mathematical terms. So here we introduce a few mathematical definitions for first the transcriptomic plasticity of tumor cells, the genetic plasticity of tumor cells, and then at the end I will briefly mention the definition of transcriptomic plasticity for immune cells. All right, so first was transcriptome plasticity of tumor cells. When we look at the traditional gene expression profiling, the issue of tumor heterogeneity here is that it prevents the quantification of cell type specific plasticity. So for example, in this simple illustration, we thought that we're observing the red versus the green, red for the pure tumor, but in reality, we're observing the orange, which is a mixture, the expression levels of a mixture of cells from the tumor and the normal cells. So when we looked into the previous work in how to separate the tumor-specific expression versus non-tumor, there are experimental approaches such as laser capture microdissection, which requires a pathologist and a very fancy, expensive microscope and a pathologist dissecting out the tumor cells versus stromal cells and send them for profiling for one sample at a time. So it's laborious and can now scale up to hundreds of tumor samples. Therefore, in silico dissection becomes a bottleneck challenge for mathematicians like us to solve. So back in 2012, and then early on, 10 years before that, people have already realized the problem. In average, one paper a year is published on trying to solve the issue. So all of the models boils down to the simple linear equation of A times X equals to B for each mixed sample, where A is a matrix of pure cell expressions with the number of genes on the rows, say 10,000 genes, and then the individual mixture component, a pure component on a column. So for example, say a mixture of 3 components, so 10,000 by 3. And X is then a vector of the proportions of these individual components, say a vector of 3. And A times X gives us a vector of expression levels, which is the observed heterogeneous expression data. All right, so how do we solve this? So previously, people thought when we're given a B, how about we require knowledge on the A, which is mixing proportions, and then we can recover the on X, and then we can recover the A. So the challenge issue with requiring the knowledge on X is that it is difficult to estimate the actual mixing proportions, and the pathologist estimate under the microscope, as I'll show you later, it does not reflect the mixing proportion at the molecular level. So alternatively, people then require knowledge on A for some genes to be able to recover the X given the B and a submatrix of A. What that means is that instead of 10,000 genes, how about we identify 200 genes, 100 genes, or 50 genes, with these genes having the fixed expression level for each of the pure component. So these are the genes that are called reference or marker genes, and I call this approach as supervised. So when we think about this approach, the main challenge is the existence of knowing the existence of these genes for each of the mixing components, which is which contradicts the plasticity of tumor cells. So for the tumor component, these genes do not exist, not in my knowledge. But this approach does work when people perform immune subtype deconvolution, because with different immune subpopulations, the reference and marker genes do exist. But our goal here is to separate tumor cells from the non-tumor cells. So therefore, we have to go with a semi-supervised deconvolution, where the full the marker genes are not available. So that is our goal. So here I use a 2-component deconvolution as to prep you use the input and an output, and then the mathematical foundation. So here, for our modeling framework, we require that the input data as first in the mixed expressions, as is shown on the right. This is actually a data set where the brain and the lung tissues of rats are used to extract RNAs, and then they're mixed in known proportions, and to generate this heat map. So each row is a gene, and each column is a mixed sample. So this is mixed expression data matrix, and as well as expression levels that are provided by one of the two mixing components. For example, here, the 100% of lung. And then our desirable output is both the mixing proportions and the individual pure expressions with genes on the rows and the sample on the columns for both the tissue one, for example, the brain here, and as well as that what is not shown, another full-size matrix for the other tissue. So the general, the mathematical framework is as simple as a linear constraint. So here for gene G and sample I, pi I is the tumor proportion for sample I. So we have YIG equals to pi I TIG plus 1 minus pi I times NIG. So here Y is the observed expression, T is the tumor tissue specific expression, and N is the non-tumor tissue specific expression. I call it normal from now on, but as we all agree that they're not normal cells, but the surrounding cells in the tumor tissue that are not tumor, such as stroma and immune cells. Although a simple linear constraint, the estimation of the parameters is challenging because it is not a finite mixture model, but a linear combination of distributions where the mixing proportion changes over samples. So the main challenge of this type of deconvolution, which is unsupervised, we do not have marker genes, and we would like to estimate a lot of parameters, are twofold: the model identifiability and the model dimensionality. So here I use a simple example to introduce the two concepts in a general term. So here say, let's simplify and then say N is known, the normal component with minimal variance. So we can define YIG star equals to YIG minus NIG. So for each gene G, then we have YIG star equals to pi I times TG minus NIG. So here, with N samples, we have N plus 1 parameters to estimate, but only N observations. So there are multiple solutions for these equations. And similarly, when we're given one sample, then we have YIG star equals to pi I times TIG minus NIG. So here we have a gene G plus 1 number of parameters to estimate, but one less observations, again, unidentifiable. But luckily, there are usually a large variation between the T tumor specific expression across genes and then a variation across pi. So when we put together multiple samples and multiple genes, the matrix decomposition can provide potentially unique identifiable solutions. However, so far we have not talked about variance. So there is a variance attached to the observed YIG, and then variance attached to the TIG, and then variance attached to NIG. And that is what's difficult, and what introduces this whole parameter space as a rocky mountain as compared to one big hill to climb in terms of model-based parameter estimation. And in terms of the dimensionality, the total number of parameters to be estimated we have two steps. The first step, when we estimate the mixing proportions, as well as the mean and the variance of expression for each gene for each of the components. So we have number of genes, number of samples, 200 plus 20,000 genes times 2. And then the second step, once we have these initial parameters, is to generate the full gene by sample matrices for each of the components, which is 20,000 times 200 times 2 values that we have to estimate. So hence the high dimensionality. So we first developed a two-component mixture model, and here is the full likelihood. The only thing I want to highlight is this integration. Because of the limitation at the actual distribution of the data, we are limited. We will have to use a numerical integration for the likelihood calculation, which brings additional challenges in the identifiability and the dimensionality of the model. And therefore, extending from a two-component mixture to a three-component mixture, which is desirable because it allows us to look into the immune component specifically, which at the end I will then bring in the transcriptome plasticity score for the immune cells. But this extension took us 5 years to then be able to resolve a double integral derived likelihood elegantly and accurately. So here, again, a little bit more further on the three-component deconvolution. So with three-component, now we have yig equals to pi 1I times n1 plus pi 2 times n2 and then pi t times tig. So n1 is the immune component, and then n2 is the stromal component. The input data here include the mixed expression data, the unmatched expression profiles for p-1, so which is two components, and the output included both the mixing proportions, as is shown in this triplot, as well as the deconvolved expression profiles, as is shown in this illustration, for each of the three components. So moving from 2 to 3, the parameter space becomes even more high dimensional, and the model is more weakly identifiable. So what happened was my former PhD student, Yang Wang, so he identified a way to tease out the parameter space in an orderly way. So first, the parameter space where we have mu's and sigmas for genes are called the genome-wise parameters, whereas pi's are the sample-wise parameters. So this bipartite plot shows that these parameters have this nice independent conditional relationship against each other, in the sense that when we know all of the mu's and sigmas, then the pi estimation are independent of each other, and vice versa. And this then allows us to utilize a previously derived iterative algorithm called the iterated conditional modes, which was published at JRSSB in 1986 for solving noisy imaging data. And the benefit of finding out an algorithm having been developed is that the JRSSB provides the entire mathematical proof to demonstrate this approach has a never decreasing likelihood and is guaranteed convergence to local maximum, which is to our advantage. So we are confident of the parameter estimation. However, we were not entirely happy with local maximum, as I mentioned to you. With a rocky mountain, the local maximum is not equal to global maximum, even though the global maximum is what we desire. We therefore propose a gene set based component merging to ensure that our parameter search is located around a global maximum. So how do we do this? So with the given N1 and N2 component, we started out with a two-component mode. So we first identified genes with similar expression from the N1 and N2 component. So, and then for these genes, we merge the two components and perform a two-component deconvolution to estimate the pi t. And then we move on towards a separate set of genes where the N1 and N2 are for sure differentially expressed. And this is the session where we perform the three-component deconvolution to estimate pi 1 and pi 2. This is a simple procedure, but what it allows us to do is to avoid the local maximum if we just go straight ahead and perform three-component deconvolution with genes where they are similarly expressed in N1 and N2 component. And here's a simulation showing that the root mean squared error, the smaller the better, is much improved for pi 1 and pi 2 estimation when we use the component merging to start at the global, the neighborhood of a global maximum. All right, so so that's about the maximum amount of mass. So now we'll move into the interesting data data part of the talk. So in terms of validation, we performed various in silico mixed experiments and also a I call it Brown's validation, where we use the laser capture microdissection data of 23 mixed tumor samples from prostate cancer. So here we have profiles of both expression profile of both the mixed tumor and then the dissected the pure tumor and pure stroma for the same patient. So here is where we found that the consistency of our estimated pi-t and the pathologist estimate is almost non-existent. So we didn't see any consistency. However, because we have both the pure tumor, and the pure stroma component, what we did was to run DMXT 2 times. One time was the mixed tumor and then the pure tumor component, and the other time was the mixed tumor and the pure stroma component. And if the algorithm works well, then the summation of the two proportions should be 1, and which is exactly what we observed with DMXT. And then we are also, with the LCM data, we are also able to confirm that individual deconvolved expressions work well, as we have the actual pure tumor and pure stroma component. So we further then went to the using, as I mentioned, using this in silico mixed cell line data to validate the 3-component deconvolution. And there also an interesting story is that we first did not observe any consistency between our pi-t and the biologist estimated proportion when they performed the experiments. So we then asked what happened. We performed a similar experiment like what we did here, that we estimated the proportion of the 3-component iteratively and summed them up, and then they did sum up to 1. So then what happened was when the biologists mixed the cell lines, they counted the proportion as the proportion of the cells. So we went back and asked the biologists to recalculate the proportions as the amount of RNAs presented in the cell, and saw a perfect correlation between our estimation with the estimated RNA proportions. So therefore, that's where we first realized that DMXT estimates transcript proportions and not the cell count proportions. So then finally, that lead into our the main topic of today. So why is that? What is the biological implication? So it turns out there has been, to me, there has been interest in finding out the tumor cell features such as RNA and DNA content to differentiate atypical hyperplasia from the true malignancy cells over the past 50 years. So this like the beginning, from the beginning of the quantitative tumor cytochemistry or fluorescent in situ hybridization. However, these technologies has always been limited to low throughput, to low amount of tumor cells, or just limited amount of genes. And then more recently, the single cell RNA sequencing technology has allowed for large scale quantification of cells. And then shortly after its invention, a group of researchers at Caltech reported a significant variation in the total mRNA content across different cells by using their spiking quantification. Furthermore, various statisticians has pointed out there is a transcriptome size and a library size factor, the two of them confounded, of course, and the total of which are measured by UMI counts. So standard normalization that accounts for the variation in the total UMI has been proposed in the statistical analysis of single cell data. And early in 2020, a group of researchers at Stanford has conducted this very interesting study across more than 15 single cell data set, and pointed out that single cell transcriptional diversity can be measured by the total number of expressed genes, and which turns out to be a better hallmark of developmental potential as well as differentiation state in tumor cells than any specific gene pathways alone. They used a various machine learning approach to demonstrate this. So far, there's no systematic study about effects of total mRNA expression levels in tumor cells in human tissues at scale, specifically as a feature of cancer cell plasticity, which then is linked to clinical outcomes. So our study aims to fill in this gap in knowledge, and for further information and 50 pages of supplementary materials, please visit our bioRxiv version of the paper. And then this work is led by my postdoc Shaolin Tao and Jennifer Wang, who's a head and neck surgeon and cancer biologist at MD Anderson. So first, we looked into single-cell RNA-seq data from 9 patients in 4 cancer types: colorectal, liver, lung, and pancreatic. And 2 of these 4 are from MD Anderson. So we are able to access the detailed clinical follow-up information. So first thing we observe is that there is indeed a much higher diversity in the total mRNA expression in tumor cells versus non-tumor cells, as is shown in here. So the rows are the cells, individual cells, and the columns are genes. And I'm showing you both the gene counts, so the total number of expressed genes versus the total UMI counts per cell. And then tumor cells, darker, the contrast between the black and the gray, as well as the contrast, the range of the values of gene counts in total UMI is much higher in tumor cells versus the other cells. We then look into the, so let's zoom into the 2 patients with colorectal cancer. So here, the Seurat clustering, we performed Seurat clustering and further grouped small clusters and found 3 clusters of tumor cells presenting distinct amount of gene counts and average total UMI for example, here, patient 1. And there are also 2 clusters in patient 2. But the additional cluster of tumor cells in patient 1 presented the higher level of total UMI than any other clusters or any other cell types. We then performed the gene set enrichment analysis and to characterize these high UMI cancer cells. First, they're not the cell cycling or proliferation. They actually have lower cell cycle scores, and they are presenting ENT signals as well as enriched for stem cell signature gene sets. So this also then links to the concept, the traditional concept of plasticity. We then ask, as we're interested in, again, the tip of the iceberg, not the bottom. So what we did is we created a pseudobulk by averaging across all of the tumor cells and divided by all of the non-tumor cells and calculate this mean UMI count ratio of tumor versus non-tumor. So what we saw is after aggregating, we can see that the average signal is distinct between these patients. So keep in mind that, so here I'm showing you the progression-free survival documented for contrasting the 2 patients within the individual cancer types. With lung cancer, we only have the stage information. So the conclusion with this limited data set is that a higher diversity in total mRNA expression in tumor cells is observed from patients with poorer prognosis across cancer types. However, we are limited with a small sample size by using just the pseudobulk generated from single cell data. And hence, and therefore we hypothesize that a quantification of average tumor cell total mRNA levels in bulk samples across large patient cohorts might be useful to track tumor phenotype and provide its direct link with clinical outcomes. So why haven't anyone done it before? So there were 2 challenges we believe prevented previous studies from addressing this question. So first is was elegantly presented by Rick Young's group many years ago using the microarray data in the sense when they studied the MYC amplification is that when individual genes are increased in the mRNA level, then after normalization of the microarray or RNA-seq, then we can still observe it. But when all of the genes are increased in mRNA level, then it is fully confounded with the normalization procedure and will be normalized out during together with the technical artifacts, which is necessary for any of the high throughput sequencing technology that we're using today, and that is before the differential expression analysis. And so that's one challenge. And the other challenge is, again, the tumor heterogeneity, that the signals we're observing are not only coming from the tumor cells in the bulk sequencing data. So therefore, we propose, because of our track record on deconvolution, a solution that uses one challenge to solve the other, which is deconvolution. The essence here is that deconvolution contrasts two groups of cells within the same experimental condition, hence canceling out only the technical effects by default, while maintaining the ratio of the tumor cell to RNA mRNA expression if there is a difference. So we propose a mathematical model for tumor-specific total mRNA expression called TMS. So TMS measures the ratio of the total mRNA expression per haploid genome for tumor cells versus non-tumor cells. And to calculate TMS, we indeed have everything that's needed from the currently developed software tools, either from both from my lab and from our collaborators' lab. So why is that? Because first, remember, in order to cancel out the technical effects, we have to calculate a ratio, and that ratio of tumor cells versus non-tumor cells can be mathematically transformed and then exactly equal to the ratio of the proportion of tumor-specific transcripts versus the non-tumor-specific transcripts, pi over 1 minus pi. And similarly, the second step is that we want to calculate the per cell mRNA. So we need to adjust for the amount of tumor cells versus non-tumor cell counts, which mathematically is also equal to the rho over 1 minus rho, where the rho is the proportion of or tumor cell purity that can be estimated from DNA sequencing data. And then lastly, we are not particularly interested in the dosage effect, so we would like to calculate a per haploid genome mRNA quantity, and therefore we adjust for the ploidy of the tumor cells versus the non-tumor cells, which is assumed to be 2. So what that means is, with a given tumor sample, as long as we have the matched DNA-seq and RNA-seq data, we can calculate TMS. So we use existing tools such as ASCAN and Absolute to estimate psi t and rho, and then we use dmxt to estimate the tumor-specific to mRNA expression proportion pi, and then we calculate TMS as simply the odds ratio of the pi of rho, and then further adjust it by ploidy. And a little bit more on the analytical side here is that people are usually quite wary about the accuracy of the ploidy and proportion because there's also identifiability issue there. So we took the pain of evaluating the consensus between ASCAT and Absolute across 15 cancer types in TCGA, and found that for the purpose of TMS, 95% of them are in a reasonable agreement. So we did filter out 5% that did not agree. And further with dmxt, there is a question of how accurate it is, how precise it is in that we can estimate pi, because now we are making it a specific biological influence. So we want to ensure the highest quality of the pi estimation. So what we did here is to propose a profile likelihood approach. What that does is that because we only observe y and some of the normal reference, we don't really know what the hidden distribution of T and M. But ideally, we would like to use only the genes for estimating proportions that show one single mode distribution which are distinct in the T and N component, and avoid all the other undesirable properties presented in some genes, for example, genes that are differentially expressed in different tumor subclones. So the profile likelihood allows us to generate this gene selection score, which will be smaller for a nice profile, and therefore these genes will most likely follow the desired distribution. And we rank all genes based on the gene selection score from small to high, and then only use the genes with the small desirable scores. And these genes form a set called, we call it intrinsic tumor signature genes. And these are the genes that we then use to estimate the tumor-specific mRNA proportions. So we have deployed the TMS to more than 6,000 patient samples by now, with samples from TCGA, ICGC, UPC, Metabric, and as well as the TracerX data. I don't have time to talk about the TracerX, but I'll show you results from TCGA and Metabric. But here is an overall distribution of TMS for each of the cancer types. So from this view, we can already see that other than the head and neck squamous, TMS presents are relatively low in values for cancer types that are more indolent, whereas the more aggressive cancer types such as liver cancer, pancreatic, and lung cancer presents among the highest TMS. On the right-hand side, I'm also showing you a benchmarking study where we actually measured the RNA content using qubit and calculated the true TMS as shown in the blue dot, and then evaluated the uncertainty in TMS estimation. And compared to the inter-patient variation, the uncertainty for a single TMS value estimation is relatively low. Next, further more biology. So we ask whether tumor cell total mRNA expression is associated with known prognostic characteristics, and the answer is yes. So we looked at the HPV status in head and neck squamous and triple negative status, ER/PR, and subtype the PAM50 subtype or MYC amplification in breast cancer, the Gleason score in prostate, the luminal versus basal in bladder, and then type 1, type 2 in renal papillary. And all of these high TMS is associated with the more aggressive feature. And what's observed in the breast cancer is identically replicated in the Metabric breast cancer study. So this is the most striking result that we have observed, and then which goes back to the concept of TMS measuring cancer cell feature of cancer cell plasticity. So we found that TMS can refine prognostication on top of the pathological stages in all 13 cancer types where the pathological stages are available. So here I'm showing the forest plot where where we include the age and then main effect of TMS in black, and the purple the stage advanced versus early in purple, and an interaction term between the TMS and stage. And across all cancer types, the TMS effect there is a significant effect on the main or the interaction term of TMS. In terms of what it means in the Kaplan-Meier output, we can see that, so here is both the pan cancer and then a specific example with lung adenocarcinoma, where within the early stage or the advanced stage, we saw that high TMS correspond to a worse prognosis. Interestingly, so when we look further into breast cancer, we saw this reversed effect of TMS, as that so with both the Metabric and the TCGA, we observe a high TMS corresponding to better prognosis. And this can be further illuminated within the Metabric by looking into the chemotherapy status, in that in both the triple negative in this column, as well as the ER positive and HER2 negative group, the patients who already who have had the chemotherapy, the high TMS correspond to a better prognosis, as also shown here in the Kaplan-Meier curve. And this is consistent with the literature on breast cancer, with more aggressive breast cancer subtypes are seen to be more responsive to chemotherapy. And the other interesting observation is when we compare TMS with type DX, which is a risk score that's currently used in clinic for assigning ER-positive, HER2-negative breast cancer patients to perform chemotherapy is that within Oncotype DX high-risk group, we can identify a low TMS group of patients which may not have benefited from chemotherapy and might suggest alternative treatment options. So in summary, TMS has the potential to serve as a marker for worse prognosis as well as for better response to chemotherapy across cancers. To summarize this, the tumor cell total mRNA expression or transcriptome ploidy in analogy to genome ploidy provides a key feature to track tumor cell phenotypes. And as compared to total mRNA expression being a hallmark for developmental potential in normal cell types, tumor cell transcriptome ploidy plays a more intricate role in cancer, likely due to its interaction with tumor microenvironment and drug treatment. So I think okay, yeah, so I do have 10 more minutes to talk about the genetic plasticity and as well as to mention the transcriptome plasticity. So I'll have to, I think I'll have to make it brief. But the concept of varying transcriptome, of course, is not, and then varying genome has been going on for a really long time. And my group had the fortune of participating in the pan-cancer analysis of whole genome working group for the past 6 years. And then specifically, we were a member of there are 17 working groups in total, and with different aspects of data analysis with the goal of understanding 2,600 cancer whole genomes across 38 cancer types. And we are part of the group studying the timing and intratumor heterogeneity. So as part of a member of this group and then the wider PCOG, we aimed at developing statistical methods for the accurate measurement of high-throughput sequencing data, as we first developed a muse for subclonal somatic mutation calling accurately. And then once the subclonal mutations can be identified accurately, we then developed a CLIP for subclonal reconstruction accurately and quickly. And CLIP has the advantage of being about 100 times to 200 times faster than the Dirichlet process-based methods, which is the mainstream of subclonal reconstruction methods until CLIP comes along. We then developed a CSER for consensus subclonal reconstruction because of the seemingly chaotic or too hyperactive field of method development for subclonal reconstruction and the challenge of not able to validate the result of subclonal reconstruction in real data. So, and then I think I'm just gonna skip the results session and apology for that. So and then jump right into a conclusion that we propose to measure genetic plasticity using subclonal tumor mutation burden and using CLIP, which is much faster than any other available subclonal reconstruction callers. We then were able to sift through the entire PCOG 2,600 samples, as well as the entire TCGA, more than 9,000 whole exome sequencing data to evaluate the performance of, or the predictive performance of the subclonal tumor mutation burden. And using tumor mutation burden to evaluate the clinical outcome is not a new idea because of one specific biological pathway is the new antigen response, which is particularly should be particularly reflected when we evaluate the subclonal tumor mutation burden in whole exome sequencing data. And lastly, we came back to this three-component deconvolution using DNXT, where we can estimate a proportion for all of the immune cells aggregated, and then as I mentioned repeatedly, and then how we use DMXT in estimating TMS, this pi mixing proportion for the immune cells refer to the amount of transcripts that are expressed by all immune cells in aggregate. We looked into prostate cancer, and we found this immune transcript proportion can be used to separate patients with distinct progression-free probabilities over a 10-year follow-up. And this is, by the way, this is TCGA data. What we did was simply to dichotomize the patients at the median of immune proportions, and then take all of the patients disregarding their Gleason score, either 3+4, 4+3, or even 8+, and then found that none of these patients developed had a relapse over 10 years of follow-up. So we believe that the transcript, the varying total mRNA amount is not a unique tumor cell specific behavior, and therefore propose to evaluate it, also the immune cell populations and in future fibroblast cell populations using our transcript proportion estimate from DMXT. Lastly, the immune plasticity also tracks tumor cell plasticity and we have some preliminary results showing the clear interaction between the two measurements. To summarize, we have developed deconvolution models to help define features of tumor plasticity in mathematical terms, and in order to improve the genomic definition and therapeutic strategy for heterogeneous tumors. And I have focused today's talk mostly on the transcriptome, measuring transcriptome plasticity in tumor cells, and briefly on measuring genetic plasticity of tumor cells, and ended with a teaser and then more work to be done on the transcriptome plasticity for immune cells. And all of the results that I presented are based on publicly available R packages that were, or software tools that were developed by my lab. So please feel free to use them and contact me if you have any questions. And then this is my team, and then my clinical and then biological collaborators, and George is very new on the list as we we are also looking into the total the other RNA species in terms of their measuring their total amount as a representation of plasticity. And thank you. I'll end here and then open for questions.
Thank you very much, Mrna. It's very interesting. So basically, behind apparently very simple and very important question, it's a lot of hard mathematics and hard thinking. So let's start with the questions. So Mrna Singh, which one is more relevant in prognosis? Total messenger RNA level or messenger RNA diversity? Can such information at an individual patient basis be useful in choosing therapeutics?
Yes, I mean, that's we're also curious about it, and then definitely so total mRNA expression is playing a essential role in the sense that the prognostication effect we observe across so many cancer types. I've never seen any other report on a specific gene or mRNA that's that can do the same thing, right? So, and if you the next steps, of course, is to dig into each individual cancer type, which is why I'm recruiting. I need more people to work on this, one cancer type at a time, where we can use machine learning tool to incorporate all of the possible signals for that cancer type, and then to further tease out their interaction or causal relationship. That's the upcoming step. And then in terms of mRNA diversity, that is, if you're talking about, sorry, just one sec, oh, sorry. If I understand your question correctly.
Yeah, can such, please.
Yes. So the diversity that when we observe diversity, it's a variance, variation at the single cell level, and then we at the bulk samples, then we will have to, we're limited to an average value. So an analogy to this would be in mathematics. It was Poisson distribution. The mean and variance are identical. And in other exponential distributions, the mean and variance are highly correlated. So the whole point of this actually can work, which is surprising, is that the mean measurement in the bulk seem to be able to track the mRNA diversity in the single cell, whereas the single cell data, we do not have enough single cell data to associate with clinical outcomes, but we do with the bulk sequencing data.
Thank you. Another question, why TMS is a better index than total messenger RNA output for clinical phenotype?
Say again?
So why TMS, the score, it's a better index than total messenger RNA output for the clinical phenotype?
There's no, as far as I'm not aware of any other way to measure total mRNA phenotype at scale. Because so we use TMS to scan through 6,500 patients, and then be able to reach this pan-cancer feature of total mRNA expression. This is not, we're not saying that you can painstakingly measure total mRNA with, I don't know, one sample or two samples, right? It's just a different scale, and then we need a mathematical measurement for at-scale evaluation.
Thank you. Now, other question, a little more technical. So, two questions in the same comment. Basic question, gene expression are in what format in order to analyze your score? Images from chip or what format? This is the first question. And the second, linear constraint for distinguish normal cell and tumor cell. Please introduce a little more about your model. Many thanks. So this is Juan Bayer. I think you can also discuss by email with Dr. Bayer at
probably that's, we probably email. So in what format to announce? But this is RNA sequencing data. So if you talk about RNA, all the sequencing high throughput, they started out with images, but I'm not sure if that's what Juan is asking, but happy to talk offline about it.
Thank you. Now let's go with one more question. So Sanjay Singh is saying, Dr. Wang, thank you for your presentation. My question relates to the problem of correlation versus causation of gen sets expression profiles to tumor growth behavior. Now the question, are there any ways to find causal links in silico? Do you factor in known interaction, positive or negative regulations between genes in a given total messenger RNA expression profile?
So total mRNA means a summation of RNA across all genes, yeah?
Yes, and he's asking about do you factor in known interactions between genes when you measure the total messenger RNA expression profile?
No, it's a summation of all. So it's only, we're saying that it doesn't matter how they regulate, how individual genes interact. It's really the summation of all mRNA content that itself is a feature. of cancer cells.
And one more question, because it's very clinical. It's to our clinics. Where's the transcriptome ploidy trends consistent between post therapy among different cancer types? So otherwise, if the therapy affects this transcriptome ploidy, and I like these terminologies that you introduced, transcriptome ploidy.
I love it. Thank you. Yeah, sorry. So say the question again.
So if the transcriptome ploidy correlates with the pre-post therapy among different cancers that you analyze.
Yeah. So we're always open to people sharing their data. So far the well documented the treatment data is only available from Metabrook in all of the sample data that we have gotten, and I believe I've shown that it is associated with response to chemotherapy as a first step. Of course, it's just one study, and then we would love to expand our evaluation in other data in other cohorts.
Thank you very much. So I think with this one, because time it's important. Thank you very much, Weni. For everybody who has question, bring contact Dr. Wang directly. And with this, thank you all for being with us. We will come back at start of September with Dr. Vinay Puduvali, another very interesting presentation. And with this, happy 2022 to all of us. Thank you, Weni, for being with us, and enjoy science and make discoveries, all of you. Thank you very much, and good luck with everything.
Thank you, George. Happy 2022, everybody. Thank you.